All right, hello everyone. Welcome to week 10 of our Bio 161 lab. This week we're going to be talking about fly genetics. It's going to be pretty similar to the plant genetics experiment we did in the past with some small changes. So you're going to be able to go over some of the statistical analysis, for example, again. And then we're also going to talk about the third part of our DNA barcoding lab. And I'm going to tell you about DNA sequencing, and that'll be cool because we'll be able to synthesize a few principles that I've taught you about in the past in this lab. So that should be interesting. Okay, so for this fly lab, if you haven't already looked at the content page in detail, we're going to be performing a cross with flies that is the same kind of cross that we did with the plants. So what I mean by that is that there's two parents that are true breeding, so they're homozygous for their traits. And then you're going to find two traits in the flies that you want to study. And you're going to do that by just looking at the picture of the parents we provided. And there's two traits that should be pretty obvious that are different between the mother and the father. So because they're homozygous for all of their genes, aka true breeding, we can make some predictions about what their offspring will look like. So they're homozygous, so that means it should be pretty easy to predict what their offspring will look like because we know which allele each parent will donate because they only have one allele for each gene because they're homozygous. Then once we have a prediction of what the F1 phenotypes and genotypes will be, we can pretend that those individuals then mate, and we can make predictions about this next generation, the F2 phenotype and genotype. So again, the parents are homozygous for the traits of interest, like in the plant lab. This time we're working with flies though. But this time you're going to be finding these traits yourself. Also like the plant lab, we're going to make predictions about the F2 generations using Punnett squares. So one slight difference between this lab and the plant lab is that you're going to be investigating sex linkage instead of just the kind of linkage we studied in the plant lab. So you're going to set up your Punnett squares a little differently for this. And it should be pretty intuitive because we're going to be pretending there's this trait that, or a gene rather, that's only on the X chromosome and is not on the Y chromosome. So therefore the XX genotypes, the females, will have two chances to pass on their alleles, whereas the males only have one chance to pass on the alleles. And we represent that by only putting the allele on the X chromosome because that's the only chromosome that it's on. Then we leave the Y blank, and then we can see the resulting offspring. So in this case, half are male, half are female. And then you can also make predictions about the phenotype based on what each allele is. So the first thing you're going to be doing is making predictions about the F1 and F2 phenotype. And you're also going to want to take into account this sex linkage. So after you've made your predictions with the Punnett square about the ratios of the phenotypes and therefore the amount of flies in each phenotype, you're going to test your prediction against the data provided like you did for the plant lab. And this will be another opportunity to exercise your statistics muscles a little bit. I think everyone will eventually take a statistics class, and it's super useful skill for understanding data in general for the rest of your lives, honestly. So I'll give a quick reminder about some of this like fundamental statistics terminology. So chi-squared analysis is just one basic statistics tool. And the whole array of statistics tools that you'll learn about if you take the class give you a p-value. And the p-value stands for probability because it's the probability that the two data sets you got are different only by chance. So for that reason, if it's a very low p-value, we can be confident that they're truly different and it was not just random. So we kind of arbitrarily say sometimes that 0.05 is our threshold, and that if our p is under 0.05, then we're over 95% sure that the two groups are actually different and it was not just by chance. So again, the chi-square analysis will give you this p-value, and then it's up to you to use the p-value to interpret the data sets. Okay, so that's actually all I have to say about the flies. This is going to be a very short lecture. It's definitely more of a self-led investigation this week. And I'll also be sticking around for those of you that are here for an hour after this class for my office hours. So you can maybe have a chance now to look at it and ask me any questions if you have them. But I'm going to move on now to the DNA barcoding project. So last time we talked about gel electrophoresis, and as a reminder, I think most of you understand this by now, but gel electrophoresis we used to measure our PCR product so that we could validate that our desired gene was the one that was amplified because we know the size of the gene. So now we have validated our PCR product. We're sure we have a tube full of that concentrated barcode or that sequence of interest. Now, how do we go from a tube full of DNA to actual A, C's, T's, and G's? That's DNA sequencing. And that's obviously a super important technique for all kinds of biological research. And there's quite a few ways of doing it, actually. But we're going to talk about one of those ways that's still sometimes used. And I also really like this because it'll remind us about some of the principles I've discussed in the past. Okay, so for Sanger sequencing, what we're doing is we're going to be using these NTPs like we did for PCR. So that's the nucleotides that get assembled into a DNA chain. However, in our Sanger sequencing reaction, some of them will be these DD NTPs, which basically just has a slight chemical modification. And you can see it up here, I believe. Not that chemical modification really matters for our purposes, but what it results in is that the new DNA strand being synthesized is terminated. So because there's a chemical change on the DD NTP, the next nucleotide cannot be added, and the full strand will not be made, and it'll be cut off short. So the NTPs, the nucleotides, are randomly incorporated because they're all just floating around in solution. And every once in a while, these DD NTPs will be incorporated instead, and it will terminate the synthesis of the new chain early. But it's important to note that not all of the nucleotides are these DD NTPs. Some of them are just the normal nucleotides. Okay, so hopefully you can imagine now that means we now have a whole array of DNA strands of different sizes based on if the DD NTPs got incorporated or not at a certain point. So next important thing is that each DD NTP of each type, either A, C, T, or G, is tagged with a different color. And this is an example where fluorescence comes back in as a super important tool, because each DD NTP, as you can see here, is tagged with a different color. And because it's fluorescent, when we shine a laser on it, it will emit light back of the appropriate color. So this allows us to detect which DD NTP was randomly incorporated. Was it an A, a C, a T, or a G, based on the color that is emitted. So now we have a random soup of our DNA strands. And by the way, this is our barcode at this point. So we're basically doing PCR on our barcode to make even more of those strands. But this time, not every strand is going to be full-length barcode. Some of them will be cut short because some of these DD NTPs are included. And because they're labeled with colors, we can see, was it an A, a C, a T, or a G that was incorporated to cut it short? Okay, so now we run this mixture of different DNA strands with either A, C, T, or G, DD NTP at the end through basically a gel like we did before. They call it a capillary gel because of the shape, but it's really the same thing, where smaller strands are able to travel further through. So now we've effectively separated all of our strands by size so that, let's say, one strand was being made where we had one nucleotide incorporated and the second nucleotide was a DDNTP. That's going to be an extremely short one. That's only going to be 2 nucleotides long. So that will go all the way down to the bottom. Next, what we do is we run this capillary down through a laser. And the laser will excite the fluorophore using fluorescence and emit a light that tells us, was it the A, C, T, or G that was incorporated that cut it short? And in that way, it'll actually tell us the order of the nucleotides, which is our DNA sequence. And that's because, let's say the first one that comes through is the shortest and it tells us there's a T. We can assume, okay, a T is here. The next shortest maybe is 3 nucleotides and it's a G. We can say it's a G here. The next shortest is 4 nucleotides. It's an A. We can say it's an A here. And in that way, we build our entire sequence. So it's a little complicated. This is actually something that you will learn about again in mole bio and bio 475. So this is definitely not an easy thing necessarily to understand. I definitely think everyone in this class can understand it though, if you look at this diagram and maybe rewatch what I'm saying here. So great way to refresh the things we've taught you in bio 161. Also really cool thing to understand. So that's all I have for you. Super short. I'll leave this recording going in case anyone has any questions, and then I'll also turn it off after if you want to ask me questions and not be recorded. Okay, I'll turn the recording off then.